Hello. In this second video of chapter 4, we are going to look into the law of independent assortment and pedigrees. But before we do so, let me first remind you of the law of segregation, which is the first law of Mendel. In the first law of Mendel, the law of segregation, we say that considering one single feature, that one single feature is carried by homologous chromosomes. These homologous chromosomes are going to end up in separate cells after meiosis 1, right? So that's the law of segregation. The chromosome you receive from mom is going to end up in one cell. The chromosome you receive from dad is going to end up in a separate cell. So that's the law of segregation. Now, we are going to take a look at the law of independent assortment. That law of independent assortment applies when we are looking at more than one features, right? So we are looking at two features. For instance, in the case of Mendel, he looked at the shape of the seeds and the color of the seeds. Shape, it's one feature. Color, it's another feature, right? So to only say that. So now we are considering more than one feature. In this particular case, we are going to focus on two features. So two features are going to be and occur on two different chromosomes. Now, the chance of receiving one feature is completely independent of the chance of receiving another feature, right? Now, really here we are talking in terms of alleles, right? Chance of inheriting allele for one gene is completely independent of the chance of inheriting an allele for another gene that is on a different chromosome. That's one. Two, it's that when two genes are far apart on the same chromosome, chance that you're going to inherit these two genes independently of each other is very high. Okay? So the law of independent assortment also applies here when two genes are on the same chromosome but very far apart from each other, right? And this is made possible because of a number of crossing overs that can occur during prophase 1. So independent assortment is going to result from the random alignment of the pairs of chromosomes during metaphase 1. Now, to show this, we have here an individual, right, for the shape and color of seeds. That genotype is going to be capital R, lowercase r, and capital Y, lowercase y. This individual is round and yellow. This is a D hybrid individual. D hybrid. Why D hybrid? Because you have one hybrid, two hybrid. Okay? So now, the question we are going to ask is, what are the possible gametes that can be formed from one individual, considering that they are dihybrid? Take a look at how the chromosome line up in metaphase 1 in alternative 1. When we track the division, we are going to see at the end of the day, at meiosis 1, at the end of meiosis 1, we're going to have capital R, capital Y, capital R, capital Y here, and then lowercase r, lowercase y, lowercase r, lowercase y. Because of the way the chromosomes, the homologous chromosomes separated during meiosis here. And now, in alternate 2, we see that the chromosomes did not align the same way as they did in alternative 1. And as consequence of that, we are also going to have different outcomes possible here. So capital R, lowercase y, capital R, lowercase y, lowercase r, capital Y, lowercase r, capital Y. At the end of the day, we see that there are really 4 possibilities of gametes. The 4 possibilities are going to be capital R, capital Y, that's 1. 2, it's going to be lowercase r, lowercase y, 2. 3 is going to be capital R, lowercase y. And 4 is going to be lowercase r, capital Y. These are 4 possibilities, right, of gametes from a D hybrid. Now, if you have to cross 2 parents that are D hybrid, what would you get in the offsprings? This is what we are going to show here. Now, assuming that we are starting with a true hybrid for shape and color, round and yellow, wrinkle and green, we're crossing these 2 parents here. In generation 1, we are going to have everyone that is D hybrid. And this D hybrid, the genotype is going to be capital R, lowercase r, capital Y, lowercase y. Now, this individual from generation 1, if we cross them, we do a self-cross of D hybrid, what are we going to get? Now here, we have a Punnett square, a huge Punnett square, right? With now 1 parent on this side and 1 parent on this side. What are the 4 different gametes that parent 1 is going to produce? Capital R, capital Y, capital R, lowercase y, lowercase r, capital Y, lowercase r, lowercase y. This parent is also going to produce the same thing. And now we are going to cross the gametes. And as we cross this gamete and this gamete, we end up with an individual that is capital R, capital Y, capital Y. And now this gamete, we are going to cross it with this gamete. Now we have individuals that are capital R, capital Y, lowercase y. So we are going to do this for the entire Punnett square. So that now we are going to see what are the genotypes and what are the phenotypes. Now, focusing on the phenotypes, we see that for one experiment that Mendel ran, he noticed that in the second generation, he had 315 individuals that were round and yellow. We know the phenotype. 108 individuals that were round and green. One individual and 1 that were wrinkled and yellow. and 32 that were wrinkled and green. We know that wrinkle is recessive over round, green is recessive over yellow. So the individuals that are wrinkled and green are homozygous recessive, right? So in terms of ratio, we really see that altogether in here we have 16 individuals, right? Possible. Now, out of the 16 individual, only one is wrinkled and green. So that's 1 out of 16. Now, we notice that individuals that are round and green and wrinkled and yellow, they occur at about the same ratio. And that same ratio out of 16, it's actually 3 for each of them, right? 3 are green, which means they are lowercase y. 3 are wrinkled, which means they are lowercase r. Now, the round shape can be capital R. We don't know if it's lowercase r or capital R. We don't know that. For the yellow here, we know that they are yellow, so they carry a capital Y, but we don't know the other one if it's lowercase y or capital Y. It doesn't matter, but we know that they are going to occur 3 out of 16 for each of them. And then the most abundant, the round and yellow, are going to be 9 out of 15. So these proportions are going to occur for D hybrid cross. Now, for the D hybrid cross and more, we can also use the law of probability. That law of probability is basically the product rule. The probability of simultaneous independent event is equal to the product or their individual probabilities. In other words, if you have a D hybrid that you are self-crossing and you want, for instance, an individual that is both wrinkled and green, this means that you are looking for the chance for an individual to be lowercase r. So for here, we have lowercase r has a probability of occurrence that is 1 out of 4. Lowercase y is 1 out of 4. And now you are going to take a product of 1 out of 4 here and 1 out of 4 here, which is 1/16. And this 1/16 gives you the probability for an individual to be wrinkled and green, right? So if we go back to the Punnett square here, guess what? We had 1 out of 16 here, right? So we fall back on our feet, so we can actually show that the law of probability or the product rule here applies for D hybrid. Now, you can use that law of probability to track 3 threads, 4, 5, and so on and so forth. So here, say we are tracking the feature yellow urine or red urine, colored eyelids or normal eyelids, and finally normal fingers, short fingers. So each of the feature is going to be commanded by a certain genotype, right? So now, if we look at the gametes here, the gametes here are going to be capital B, capital H, and lowercase e, okay? Capital B, capital H, lowercase e, all right? Then the other possibility we have, it's going to be capital B, lowercase h, and capital B, lowercase e, right? This possibility. And then you are going to do the same thing for lowercase b. So lowercase b, capital H, lowercase b, lowercase e. So we are right here. And finally, we have lowercase b, lowercase h, and lowercase b, lowercase e. We are right here. So what we do with this parent, we are going to do the same thing for this parent, right? So that the gametes for this parent, sorry about that. For this parent is going to be lowercase b, lowercase h, and capital E, and lowercase b, lowercase h, lowercase e. So we are going to apply the law of probability here, so that we are going to have that chances that a child is going to be lowercase b for red urine is half. The colored eyelid, so capital H, lowercase h, is half. Short fingers, capital E, lowercase e, it's half. Now, let's combine all the features together to be red urine, colored eyes, and short fingers equates half times half times half, which is 1/8, right? That 1/8 is the probability for this event to occur, right? You can have, in order to determine this, you have a Punnett square right here, another one right here, another one right here, and you run each Punnett square independently of each other. And now you start counting the number of times this event is happening, this event, and this event is happening, right? So basically, that's how you solve the problem, okay? You have here an explanation of the different genotypes and the phenotypes. All right. So now what I have not said, it's actually brachydactyly. Brachydactyly, it's basically short hands. Brachydactyly is short hands. Short fingers, short fingers. Okay, that's brachydactyly. That's the technical term for brachydactyly. Okay, know that short fingers is dominant over normal finger size. All right, how about a pedigree? A pedigree is going to provide tremendous information to researchers and to geneticists. That's why whenever you go to see a geneticist, they're going to ask you, okay, well, do you have diabetes? Do you know if one of your parents had diabetes? Your grandparents, they will start asking you a number of questions because they want to assess the possibility that you may have diabetes or you may develop a certain type of cancer, right? So the pedigrees are very instrumental for genetic counselors. And when it comes to pedigree, the bigger the family, the easier it is to come with meaningful conclusions. And the more numerous the offspring, the easier it is to deduct what the parent were in terms of were they carrier of the feature, was that feature dominant, was that feature recessive, what was it, right? So it is possible to actually say that. So pedigrees are going to be made up of a number of symbols. These symbols are going to show relationship. Not only that relationship, but also who had a trait, who was homozygous, who was heterozygous, who was recessive, who was dominant, right? So there are a bunch of information that you can gather from a pedigree, which actually sets this this chart apart from, for instance, a genealogy tree, right? That genealogy tree only shows you the relationship from parents to offspring, but it doesn't show you the traits, right? So here we have some symbols that are used. Now between you and I will not test you on the different symbols, but I want you to know what the symbols are for your own good, right? When you see a pedigree, when you see a circle, that circle means the individual is a female. When you see a square, that square means the individual is a male. A circle that is empty, it's a normal female. A square that is empty, it's a normal male. A circle that is filled with, that is shaded, is going to be a female that is affected. A square that is filled, it's a male that is affected. If a circle is partially shaded, that means the female is heterozygous, so they are carriers. When the square is half shaded, that means the male is heterozygous, it is a carrier, okay? So here we have carriers, here we have affected individuals, here we have unaffected individuals. Now, some pedigree can show if people died or not, right? So here we have, in this case, dead female, dead male. A diamond sign, it's basically that the sex is unspecified. A stillbirth is going to have a male or female death. However, you're going to have a SB for stillbirth underneath. then the progeny are always represented by circle, squares, or diamond. Spontaneous abortion or miscarriage is represented like this, while terminated pregnancy is represented like this. Now, quite frankly, most of the pedigrees are going to show you the progeny, right? Is that progeny affected, unaffected, carrier, non-carriers? Now, the vertical lines are going to mean generation. Horizontal lines are going to mean partners. Dashed lines like this are going to mean adoption. And like this, these are siblings. Okay? And then other thing I want to show you here, it's going to be when parents are closely related. This is the case of consanguinity. Okay? So these are the few signs and symbols I want to address in a pedigree analysis. Also, finally but not least, we're going to have the Roman numbers. The Roman numbers are going to indicate generations, while the Arabic numbers are going to indicate individuals in a generation. Now, to be specific, let's take a look at a pedigree here. So here, you are going to have individuals in the parent individuals, right? One that is unaffected, the other one that is a carrier, right? And then when you look at, so these are the parents, okay? When you look at the first generation, you are going to have individuals that are heterozygous, right? Both male and female, which means that really here, looking at the parents, both sexes are affected almost equally. So this has to be the case of autosomal inheritance, right? Because both sexes are affected. Okay? So now, here, the pedigree here actually shows the marriage of first cousins. So the marriage of first cousins here. Remember, we have here the parents, right? Here we have the offsprings. The offspring of this parent. But interestingly, the children for this guy and this lady actually got married down here. This are first cousin. Anytime you have first cousin that come together, chance that a disorder is going to express in the offspring is very high. Okay? That's why you see that in this union here, you have two individuals that are affected. Okay? So, anytime individuals share the same set of grandparents, the risk of genetic disorder down the line is very high. All right. So, in the case of dominant traits, remember that dominant traits do not skip a generation. They can affect both sexes. We already mentioned that earlier. An example of a dominant trait is going to be polydactyly, right? Which is extra finger or extra toes, right? So here, if we consider the parents here with mom here that had polydactyly, we look at the offspring, so generation 1, this daughter inherited polydactyly from mom here, and this daughter inherited polydactyly from the same mom. And now, the individual here got married and developed a relationship with this guy right here. Down the line, we see that they had children that were affected, both affected. And in individuals here, we see that children are affected here, many of them. Okay? And when we go down in the next generation, we also have a number of children that are affected. So this is how dominant traits can be present in the next generations. So you see that from 1, 2, 3, 4 lines, you see that they are all, in all generations, they are affected individuals. But for recessive autosomal traits, the story is different, because in spite of the fact that male and female have equal chance of being affected, however, one generation can be skipped, right? Can be, not systematically, but can be skipped. This is the case, for instance, of albinism. Albinism, it's when an individual is not producing melanin because of one particular enzyme responsible for the production of melanin is not being expressed, or that enzyme is defective, right? So in the case of this individual here, right, when we look at the parents, here. So in this generation, we have one individual that is a carrier here, and an individual that is a carrier here. Now, the offsprings in this relationship is 2 children. None of them is albino. In this relationship here, none of the individual is albino. However, if these 2 individuals come together, develop a relationship, because they are both carriers, now they have a high possibility to now have an offspring with expressing the disorder, so that we have this individual who is albino and this individual who is albino. So they also have the chance of having an individual who is expressing the pigment, right? Now, remember, two of the parents have to be carriers to have the chance for expressing the disorder, right? Notice that there is no albino in generation 1, no albino in generation 2, but in generation 3, you have an albino. This is also true for other features such as cystic fibrosis, such as the sickle cell disease. Okay? Now, not all pedigrees can be conclusive, right? And this happens because sometimes it's difficult to tell if a trait is caused by dominant allele or recessive allele, right? So in this particular pedigree that you see here, the trait can be caused by autosomal dominant allele or autosomal recessive allele. Okay, so let's take generation 1. In generation 1, where an individual is affected here. This is the case of alopecia areata. In the case of alopecia areata, we see that mom here is expressing alopecia, right? That mom got married with a person, and the offspring here actually is going to produce two individuals, one expressing alopecia, the other one not expressing alopecia, right? Now, in this relationship here, the individual produced here express alopecia, right? In this relationship here, with this individual without alopecia and this individual with alopecia, we notice that, well, they have an offspring with alopecia. So in every single generation, we have alopecia. This is basically the sign that this is a dominant autosomal trait. But at the same time, though, we notice that this individual here that is normal, that is this individual here that is abnormal and who is marrying someone with alopecia is producing offsprings that are many of them normal, a limited of them with alopecia. This feature can very well be a recessive disorder, because for alopecia, whether you are capital A, lowercase a, so for alopecia can be caused by lowercase a. In a case of homozygous recessive, and it can be caused by a dominant allele, capital A, in which case we don't know what is here, right? So is it lowercase a, is it capital A, right? So when tracking what is happening here between generation 1, generation 2, and generation 3, it can really be either, okay? And that's why we say that this is unconclusive, right? So inconclusive pedigrees arise typically when the trait is not severe and when it doesn't affect fertility. Okay, in the case of alopecia, this is not a feature that is going to affect the vitality of an individual. That's one. Two, this doesn't affect the fertility of an individual, right? This is typical situations where pedigrees are going to be unconclusive. Now, let's talk about the conditional probability. The conditional probability are going to apply to basically predict recurrence risk of inherited condition. An example is going to be the sickle cell disease. So we are going to track here the case of two siblings. One is called Tanisha, the other one is called Deshaun. So what is the probability that Tanisha's child is going to inherit a gene that is mutated so that that child is a carrier? What is that probability? Now, Deshaun has sickle cell. Tanisha does not have sickle cell. Now, let's dive into this situation. So remember, Deshaun has sickle cell. Tanisha, who is a girl, does not have sickle cell. Now, for Deshaun to have sickle cell, their parents, here Kizzy and Hike, they both have to be carriers. They both have to be carriers. Now, as these two parents are carriers, now they can have an offspring that is affected, right? But now, the individual that is unaffected here, what is the probability that they're going to have a child or a fetus that is a carrier? Well, for her to be a carrier, she has to inherit one allele from mom or dad, right? So for this lady to be carriers, she has to inherit one defective allele from mom or from dad. And now that we know this, what is the chance that the unborn child here is going to be a carrier? So that chance is half, because mom is a carrier, right? Okay, so mom has half the chance to pass that carrier gene to the child. So let's break down things here. I already talked about many of these things here, but let's break it down. Let's break it down again. So Deshaun has sickle cell disease, right? His unaffected parents, they are both carriers. They have to be both carriers. Tanisha is healthy, but she can be either carrier or completely normal, right? Homozygous normal, okay? So these are the possibilities for Tanisha. Now, Tanisha's husband, Antoine, has no family history of sickle cell disease. If they have no family history of sickle cell disease, chance that they are going to be carrier is close to zero, right? So let's consider that. So now, having said all of this, right, let's consider Kizzy and Heike here. So the probability to be a carrier here, it's basically, here is a carrier, here is a carrier. So 2/3 probability that Tanisha is a carrier is 2/3 here, right? And then let's take a look at, no, let me start over. For the parents to produce carrier child, we have 2 chances here out of 4. So we have 2 chances out of 4, okay? And here, let's take a look at here, Tanisha and Antoine. Antoine is normal. Tanisha has a chance to be a carrier, right? Now, because really we have 2 chances out of 4 here, we know that actually Tanisha is not SS, lowercase s, lowercase s. Therefore, that chance here is being eliminated, right? And we are really saying that really the probability of Tanisha to be capital S, lowercase s, capital S, lowercase s, it's 2 out of 3 possibilities here. 2 out of 3 possibilities here. And now that we know that, let's take a look at the probability for Tanisha and Antoine to have a child with with carrier. So, assuming that she's a carrier here, then we are going to see that chances to be a carrier here is half. So 50%, right? Half here. So now, what is the chance that the unborn child is going to be a carrier? That probability, it's really the 2/3 here times the half that is right here, right? The total probability is 2/3 times 1/2, that is 1/3. Okay, that is basically how you calculate the probability, right? Finally, but not least, let's address exomes and genome sequencings that are tools that are used to clarify pedigree. Today, we have more and more the use of gene sequencing to clarify pedigree. This is very important because the pedigree, as we used to use it, is not anymore as simple, because remember that one feature is all when 1, 2, 3, that most features are not always explained by one single allele, one single gene. It can be multifactorial, it can be multiple gene, right? So basically, this exomes and gene sequencing is being used to address situations including when a child has a syndrome that has not seen before, okay, while both parents are unaffected. So the disease that we call orphan diseases, right? Many of these orphan diseases are subject to study by looking at exomes and gene sequencing. Okay, so in this case, actually, we can see children with expressing some disorders because both parents are carriers, but we didn't know this, right? The parents actually didn't know this, that they were carrier of the condition and they passed it down to the offsprings. But this can also happen because you have a brand new mutation that occurred spontaneously during the formation of gametes. Whether that gamete was a sperm cell or an egg cell, right? We have some mutations that are gain of functions, right? Where that function was not there before, but then because of the gain of a function, now we have an issue, right? And we have some disorders that are caused by loss of function, right? Keep that in mind. So typically, we're going to not just look at exomes, but also sequence the proteins, right? Looking at the parent exomes, looking at the parent genome, looking at the child exome and genome, right? To try to identify the the mutated genes and alleles. So by doing this exomic analysis and genomic analysis for both parents and the child, tremendous amount of knowledge can be acquired, right? And using DNA information is going to be tremendous, because now with all the tools that we have and with AI that we also have today, we can actually say what is in unaffected parent, right, compared to the offsprings, and basically be able to solve a number of problems. Also, by doing the exome analysis and gene sequencing, we can come up with potential treatments of diseases, right? and explain why some symptoms are developing, right? And be able to actually anticipate on solving the problem. So this is what I have for part 2 of 2. So this is where we are going to stop our conversation. Thank you for your attention.